Aloha and welcome to the meiosis video. In here we will talk about the life cycle of sex cells. So first we will be comparing chromosome numbers between somatic cells and gametes. We'll also describe the stages of meiosis, and finally we'll compare meiosis and mitosis. So let's talk about chromosomes. Now, there's two different kinds of cells that I want you to be familiar with. We have gametes and then we have the somatic cells. Gametes are often referred to as our sex cells, and in females, those are the eggs, and in males, those are going to be the sperm. Somatic cells are everything else. Sometimes they're called the body cells. Now, remember that when you were formed, you came from an egg and a sperm, and that gave you your complete set of DNA. And if we look at this karyotype down here, you can see that for chromosome 1, we have two copies. You got one copy from your mother and one copy from your father. And that's the case with all of these. So in essence, all of your body cells are going to have what we call a diploid condition, or 2N. They'll have two copies of each of the chromosomes. Now, if we had two copies in each sperm and two copies in each egg, when they came together, you would have four copies, and that's way too much. So our gametes here are what we call haploids, and they simply have N. They're going to have one of each of these chromosomes is all. So they'll have one or the other. And we'll talk about that process of meiosis and how we get it down from a 2N to a 1N condition. So unlike mitosis, meiosis has two different divisions. So we're going to go through meiosis 1 first. And in meiosis 1, it'll look an awful lot like mitosis. You'll see that during prophase, the chromosomes thicken up, the nucleus is going to dissolve away, and then we're going to get into metaphase. We're going to see a lining up of the chromosomes on the middle plate here. During anaphase, they're going to separate. We'll see that process going on here, and then finally through Telo Phase and cytokinesis, we form two cells. So let's take a little more detailed look at this process. Okay, so once again for meiosis, our story starts with interphase. And during interphase, the cell is going to be growing and making copies of its DNA and other organelles, including the centrioles, and everything gets ready for this division process. So this brings us into prophase 1. Okay, and during prophase 1, we get an interesting thing going on. We call this crossing over. So as our chromosomes are lining up, what you'll notice is down here, there's a little bit of crossing over the chromosomes, and they can actually switch genetic material. And this process is what separates the DNA from the mother strand and the father strand. This crossing over allows it to become a little bit blended, and that's why you're kind of a mixture of your parents, but also you're different from your siblings. It's because of this crossing over process. Now, you'll notice that the nuclear membrane is all gone. We're forming our spindle fibers here, and this is what we see going on during prophase 1. In metaphase 1, as always, we see that our chromosomes are going to line up here in the middle of the cell. Remember, M is for middle. We have our spindle fibers here. They're going to attach to our different chromosomes, and we can see that the process is getting ready to divide them up. Now, in anaphase 1, you'll see a little bit of a difference. We're not breaking the sister chromatids in half. Instead, what we're doing is we're transferring the whole chromosome to the poles. Okay? In mitosis, we would have split these in half, and each half would have come there. But here, we're going to ultimately make it one set. So we can start seeing some division here going on. Okay, and the end of meiosis 1 is with Telo Phase 1, and in Telo Phase 1, you'll notice that the spindle fibers are all gone here. You'll notice that the nuclear membrane is starting to form, and we've also had cytokinesis, so we've split into two different daughter cells. So you can see we went from one cell to two cells, just like we see in mitosis. Now we enter in meiosis 2. Meiosis 2 is what separates us from mitosis. Remember, mitosis was just one division. Meiosis is two divisions. So here, our paired chromosomes are going to start lining up again. Right? During metaphase, they'll get on the middle plate. During anaphase, we're going to separate the sister chromatids here, and then we'll have cytokinesis and telophase going on, and we'll end up with 4 cells. So let's take a look at these steps a little bit more in detail. So here we see prophase 2. Notice it's in meiosis 2, so now we have the 2 with it. And once again, our centrioles are splitting out. We're starting to form the mitotic spindle. The nuclear membrane is all gone, and we see a thickening of the chromosomes again. Also, take notice that now, instead of having one cell going through the process, we have our two daughter cells going through the process. And because these are the daughter cells and we separated the chromosomes, each of these daughter cells is going to have half the number of chromosomes as the original cell. Now we're in metaphase 2. Remember, M is the middle, so we'll see along the mid plate of the cell, or the metaphase plate, we're lining up these chromosomes, and they're getting ready. They're attached to the mitotic spindle, and they're ready to divide. Now here in Anaphase 2, you'll notice that we've attached to the centromere and we've split the sister chromatids. So now each cell is getting a chromatid instead of a pair. So the chromosomes, we had the homologous pairs where there were two together and we split those pairs up. So we had a chromosome in each daughter cell. Now we're splitting those into the separate chromatids. And that takes us to Telo Phase 2, where we start forming a new membrane. We notice that we're losing our mitotic spindle. We'll go through cytokinesis, and now what we result in is 4 haploid cells. So each of these 4 cells is going to have half of the DNA. Instead of having a full set, it only has a half set. And that's going to be important because if I take a half set and a half set and add it together, I get a full set, and that's what we need in our somatic cells. Okay, so here's a real quick summary of meiosis. Remember, meiosis has 2 different phases, meiosis 1 and meiosis 2. During prophase 1, we have our sister chromatids that are going to shrink up and get ready to go. Our spindle fibers are going to attach to the centromeres, and we have our crossing over going on at this point. That takes us into metaphase 1, where we're going to have the tetrads line up randomly in the center of the cell. Then we go to anaphase, where the homologous chromosomes are going to separate and they'll move to opposite ends of the pole. And then we go into telophase 1, where the nuclei reform and the cytoplasm divide in half. So now we have 2 daughter cells. Each of the daughter cells has half the genetic material as the original cell. Then we go into prophase 2, where the spindle fibers are going to attach to the sister chromatids. These sister chromatids will line up in the center of the cell, and then during anaphase, these sister chromatids separate and they move to the opposite sides. We get 4 nuclei forming. Cytokinesis creates 4 cells, and all of those cells are going to be in the haploid condition. Okay, so the difference between mitosis and meiosis: mitosis is in body cells, meiosis is going to be in these gametes or sex cells. Mitosis results in 2 diploid or 2 end cells, and meiosis results in 4 haploid or just a single end cell. Okay, so the lessons, will go into it in a little more depth so you can understand what's going on. And as always, good luck on the lessons, and we'll see you in the next video.